Okay. Hello, everyone. So I'm the moderator of today's seminar. So I'm Shan from USTC. So the first speaker of today is Dr. Xiaolin Wang, a postdoc from my lab. But he's going to promote to associate research professor soon. Xiaolin will talk about mammalian conserved circular RNA. Okay, and Xiaolin, you can start now.
Okay, thank you. Hello, everyone. I'm Xiaolin Wang from the University of Science and Technology of China. It's my honor to present my work at RNA Collaborative Seminar. My report title is A Mammalian Conserved Circular RNA CircLARP1B regulates hepatocellular carcinoma metastasis and lipid metabolism. CircLRNAs are widely expressed in eukaryotes. There are four subclasses: exonic CircLRNAs, exon-intron CircLRNAs, intronic CircLRNAs, CIRNAs, and mitochondrial encoded CircLRNAs, mecRNAs. Reverse complementary sequence and RNA binding protein RBPs are known to regulate CircLRNA biogenesis. CircLRNAs play important roles by acting as microRNA-bound transcriptional regulation, protein decoy, or even encoding proteins. CircLRNAs play important roles in human diseases. Take hepatocellular carcinoma HCC as an example. CircLRNA participate in CircLRNA proliferation, EMT pathway, metastasis, and drug resistance. Lots of CircLRNAs with identified functions in cancer are not conserved in mammals, which in some way hampers in deep-sea investigation for CircLRNAs with animal models. Our scientific question is to identify mammalian conserved CircLRNAs in human disease and to investigate functions and mechanisms in human and mice. First, we collected 3 metastatic HCC specimens and 3 non-metastatic HCC specimens and performed RNA sequencing. By informatic analysis, we identified 48 CircLRNAs were dysregulated, among which 5 CircLRNAs are conserved between human and mice. CircLARP1B was the only and most differentially metastasis upregulated candidate among the 5 CircLRNAs. CircLARP1B consists of exons 2 to 4 from the LAMP1P gene and a deduced size of 294 nucleotides in both human and mice. Then we performed a northern blot to verify the existence and full length of CircLARP1B in human and murine cells. Single molecular fish and nucleus cytosolic fraction revealed that CircLARP1B predominantly localized to the cytoplasm. Furthermore, we evaluated its clinical significance. With HCC specimen, we found that the CircLARP1B levels was higher in metastatic HCC specimen. Higher CircLARP1B levels were positively correlated with advanced TNM stages and predict short overall and disease-free survivals. Collectively, CircLARP1B is a mammalian conserved CircLRNA related to HCC metastasis. Then we performed loss and gain of function assays to explore the effects of CircLARP1B on cell evasion, as evasion is directly related to metastasis. RNA and knockdown or overexpression assay demonstrated that CircLARP1B promoted cell evasion. We also generated CircLARP1B deficient cells with CRISPR-Cas9 technique to delete intronic allo elements. Allo elements belong to reverse complementary sequence known to facilitate circRNA biogenesis. The LARP1B deficient cells showed decreased CircLARP1B level, but not changed LARP1B mRNA level. When CircLARP1B deficient, the cell evasion was significantly reduced. We then tested the potential metabolic alterations in CircLARP1B deficient PLC cells. Metabolics and lipidomics showed that lipid metabolism was the most enriched pathway. Upon CircLARP1B deficient, components of lipid droplets such as PC, PE, and TG performed with significantly lower levels. Lipid droplets examined by near-red staining were markedly decreased when CircLARP1B deficient. Then we asked how CircLARP1B regulated lipid metabolism. We performed a carbon-13 labeled targeted metabolic flux analysis to assess the roles of CircLARP1B on FAS. A total of 17 kinds of fatty acids were labeled, and all of them were significantly reduced in CircLARP1B deficient cells. Labeled 2 carbon units from labeled glucose and glutamines into palmitate revealed de novo FAS. Expectedly, the distribution of labeled palmitate isos was significantly lower when CircLARP1B deficiency. We further evaluate fatty FAO with OCA acids and found that the OCA rate was unaffected. Then we evaluated the various FAS and FAO associated regulators. Phosphorylate AMPK, phosphorylate ACC1, and phosphorylate raptor levels were significantly increased upon CircLARP1B deficiency. We focused on AMPK and ACC signal owing to the fact that ACC1 is the rate limiting enzyme for FAS. We wondered whether CircLARP1B promoted HCC cell evasion while modulating lipid metabolism. FASN is the human lipogenic enzyme for de novo FAS. Upon CircLARP1B deficiency, FAS overexpression enhanced lipid droplets and cell evasion. We found that FASN inhibition with small molecules blocked the promoting effects of CircLARP1B on lipid drop formation and cell evasion. Taken together, CircLARP1B stimulated HCC evasion through remodeling lipid metabolism and promoted FAS while modulating the AMPK-ACC signal. To direct investigate the physiological roles of CircLARP1B in animals, we use the CRISPR-Cas9 to generate a CircLARP1B knockout mice repeat sequence in intron 4 of murine LARP1B gene was deleted, but functional elements such as the splicing site remains unaltered. CircLARP1B mice performed unaltered LARP1B mRNA levels. In physiology, compared to the wild-type mice, CircLARP1B knockout mice had significantly reduced lipid droplets in livers. The phosphorylate AMPK and phosphorylate ACC levels were also significantly increased in the CircLARP1B knockout mice liver. We then investigated the in vivo roles of CircLARP1B in HCC metastasis. With an induced HCC mice model, we found that CircLARP1B knockout mice inhibited fewer and smaller liver tumors and lung metastasis compared to the wild-type mice. For liver tumors, we also examined the pro-metastasis marker Vimin and the anti-metastasis marker ecadherin. Vimin was significantly lower and ecadherin was significantly higher when CircLARP1B knockout. Then we examined the markers AFP, ALT, and AST. Their serum levels were significantly lower in CircLARP1B knockout HCC mice. We also tested the lipid classes TG and TC levels in serum and found that the levels were significantly lower in CircLARP1B knockout HCC mice. In HCC model, CircLARP1B knockout contributed to significantly fewer lipid droplets, higher phosphorylate AMPK levels, and higher phosphorylate ACC levels in liver tumors. Furthermore, we infused liver-directed AAV expressing shRNA against CircLARP1B into wild-type HCC mice, consistent with the results from CircLARP1B knockout mice. AAV SH CircLARP1B injection resulted in fewer and smaller liver tumors and lung metastasis. Wild-type HCC mice infused with AAV-SH CircLARP1B inhibit significantly fewer lipid drops and markedly higher phosphorylate AMPK levels and phosphorylate ACC levels. TG and TC levels in serum were also decreased. Taken together, lower CircLARP1B levels were unfavorable to HCC progression, lipid accumulation, and metastasis in mouse models. Then we set out to elucidate the functional mechanism of CircLARP1B. Due to CircLARP1B localized to cytoplasmic, we performed RNA pull-down with the cytoplasmic fraction of PLC cells with a biotin-related oligo against the backspliced junction of CircLARP1B. The complex was separated with SDS patient followed by liver staining and MS to disclose the specific binding band. ncRNPD was identified as the CircLARP1B interacting protein. CircLARP1B pull-down and ncRNPD RIP assay further verified their interaction. We also used the mouse liver as an experimental material. Pull-down and RIP assay also verified their interaction in mice. Therefore, CircLARP1B interacts with ncRNPD protein in both human and mice. We performed the colocalization experiments for CircLARP1B and ncRNPD in PLC cells. We observed that more than 70% CircLARP1B signal overlapped with HRMPT protein signal in cytoplasm. Then we used the RBP map tool to predict the binding site and uncovered 2 motifs conserved in human and murine CircLARPs. A mutation of either decreased their interactions. Mutation of both motifs abolished the interactions. We then set out to investigate the roles of HRMPT binding in the roles of CircLARP1B. Overexpressed wild type, but not the double mutant motifs could rescue the phenotype of CircLARP1B deficient, including cell division, lipid drop formation, phosphorylate ACC levels, phosphorylate AMPK levels. We concluded that the HRMPD and CircLARP1B interaction was essential to regulatory function of these CircLARPs. To provide insight into the mechanism of CircLARP1B and HRMPT in HCC, we performed HRMPD RIP with cytoplasmic fraction of PLC cells. We identified 216 mRNAs with contrast changes of HRMPT binding on the 3' UTR upon CircLARP1B deficient or overexpression. As cytoplasmic HRMPT regulates mRNA stability by targeting 3' UTRs. GO analysis for these targets illustrate biological processes such as peptidal THR phosphorylation, protein stability, mRNA metabolic process. In peptidal THR phosphorylation was the most enriched progress, among which LKB1 showed the most HRMPD binding signal on the 3' UTR. LKB1 is well known to directly catalyze the phosphorylation of AMPK, which is required for AMPK activation. We then directly overexpressed the LKB1 in HCC cells and found that LKB1 overexpression nearly abolished the effects of CircLARP1B on phosphorylate AMPK and phosphorylate ACC1 levels. Overexpressed LKB1 also blocked the effect of CircLARP1B in promoting cell invasion and lipid formation, lipid droplet formation. Then we explored the roles of HRMPD on LKB1 mRNA. HRMPD knockdown rescued resulted in significantly short half-life of LKB1 mRNA and lower LKB1 protein levels in human and mice, indicating that HRMPD stabilizes LKB1 mRNA. HRMP is reported to bind to the 3' UTR and interacts with the translation initiation factor EIF4G1 and cap binding protein EIF4E to form a ternary complex to stabilize this mRNA. We performed the EIF4G1 and EIF4E rate followed by RT-PCR with specific primers to detect the LKB1's 3' UTR with or with not HRMPD knockdown. HRMPD knockdown resulted in decreased binding signal of both the EIF4E, EIF4G1 to the LKB1 3' UTR, indicating that HRMPD enhanced the mRNA loop and stabilized LKB1 mRNA. Upon CircLARP1B deficiency, or knockdown, LKB1 mRNA stability was increased, and the protein level was also increased. Collectively, CircLARP1B competes with LKB1 mRNA for HRMPT binding and leads to LKB1 mRNA instability. We further examined the involvement of LKB1 in the effect of CircLARP1B on HCC metastasis. Liver-directed LKB1 silencing nearly restored the CircLARP1B knockout mediated suppressive effects on the serum level of AFP, ALT, AST, TG, and TC. We found that liver-directed LKB1 knockdown led to more or larger liver tumors, larger lung metastasis compared to the control in CircLARP1B knockout HCC mice. LKB1 depletion restored less swimming, more equilibrium, and less lipid droplets, more ACC levels, more phospholipid AMPK, phospholipid ACC levels caused by CircLARP1B knockout. Therefore, LKB1 was the key factor for the molecular regulatory pathway of CircLARP1B in HCC metastasis and lipid metabolism. Finally, we proposed a working model. We identified a CircLARP1B, HRMPD, LKB1, AMPK regulatory pathway in HCC metastasis and lipid metabolism. And this work was published on Advanced Science. CircLARP1B is a mammalian conserved circRNA identified in HCC metastasis. CircLARP1B regulates HCC metastasis and lipid accumulation via FAS. Murine CircLARP1B deficiency impacts HCC metastasis and lipid accumulation. CircLARP1B destabilizes LKB1 mRNA while perturbing HRMPD. LKB1 is the key factor for the effect of CircLARP1B in HCC mouse model. We thank Professor Shan, Dr. Jinxin Li, Dr. Shuhui Chang, Dr. Liang Shi, Dr. Boqiang Liu, Dr. Zhiyong Shen, and Professor Xiujun Cai. We also thank the National Nature Science Foundation of China for funding supporting. Thanks a lot. That's all.
Okay, let's see. Do we have questions? No, I don't think I don't see any question. So okay, thanks, Xiaolin. So I think now we move to the next speaker. Xiaolin, you can, close your PPT, stop sharing. Yeah. So our next speaker is Dr. Shouhong Guang. He's a professor in School of Life Sciences, USTC, and he's going to talk about small RNA biogenesis in C. elegans. So, Shouhong.
Okay, can you see me? Can you hear me and see my slides?
Yeah.
Okay, great, thank you. Okay, hello everyone. It's my great pleasure to have a chance to give an online talk here. In this talk, I will show you our recent work that germline granule compartments could coordinate small RNA biogenesis in C. elegans. We all know that RNA interference, or RNA, is a widely conserved gene regulation mechanism. When a cell is subjected to double-stranded RNA, the genes with complementary sequence will be inhibited. That is also called be silenced. For example, here is a worm. It expresses a green fluorescence protein GLP. When this worm is treated with GLP double-stranded RNA, the green fluorescence disappears. The RNA mechanism is conserved from plant, yeast, worm, fly, mice, and here human being. Especially small RNA drugs based on the RNA mechanism have been approved by FDA and used to treat human diseases. This slide just shows you a simplified model of RNA. Basically, there are multiple inputs of the RNA machine into the RNA machinery. These inputs include double-stranded RNA from in vitro synthesized double-stranded RNA, from viruses, transposons, microRNA precursors that has a hyphen structure, bidirectional transcription of the genome, and RNA-dependent RNA polymerases. All of these inputs produce double-stranded RNA that can be recognized and cleaved by a conserved ribonuclease called dicer to generate 21 to 23 nucleotide non-small interfering RNA, that is siRNA. siRNA bind to conserved to proteins in another conserved family called agalate, and then perform their downstream silencing effect. For example, they can inhibit translation, degrade targeted mRNA, induce chromatin modification, or inhibit transcription elongation. Eukaryotes could generate many kinds of small RNAs in the cell. Besides dicer-cut double-stranded RNAs to siRNA, there are additional ways to produce siRNA. For example, in C. elegans, there is a mechanism called RNA amplification to generate a large number of small interfering RNAs, namely 22G RNA. This kind of siRNA are 22 nucleotide long and start with 5' and G guanosine. Interestingly, in worms, there are two kinds of 22G RNA pathways, including the Vigo class 22G RNA and the Caesar-1 class 22G RNA. These two 22G RNA pathways play distinctly different biological roles. C. elegans expresses 4 RNA-dependent RNA polymerases, that is RDRP, to amplify RNA. These RDRPs include RF1, RF2, RF3, and EGO1. Most C. elegans 22G RNAs are synthesized by RF1 and EGO1. RF3 is required for the biogenesis of 26G RNA, and little is known about RF2. In this talk, I will focus on EGO1, which is a germline expressed RDRP to synthesize a subset of germline 22G RNA. To investigate the expression of EGO1 and RF1, we first used the CRISPR-Cas9 technology to lock in GLP tags or mCRNA or tag RFP tags into the endogenous EGO1 or RF1 gene loci. After that, we first test whether the GLP or RFP tags may disrupt the endogenous functions of EGO1 or RF1. Actually, the target animals exhibit a similar feeding RNA response and similar brood size to that of wild-type animals. Therefore, the epitope tagged EGO1 and RF1 could encode functional proteins and recapitulate their endogenous functions. We then visualize the expression and subcellular localization of EGO1. EGO1 was only expressed in the germline, but not expressed in the somatic cells. Shown here is the elegans germline, start from the mitotic region, transition zone, early and middle packeting, late packeting, diploiting, oocyte and until sperm cycle. From mitotic region to the late packeting, we can find that EGO1 accumulated in granules in perinuclear region. We will know that many biomolecular condensates have been identified to play important roles in biological processes. For example, the nucleolus is presented widely in most organisms, which is essential to express ribosomal RNA and assemble ribosomes. Another example is the Germ granule. Germ granules are RNA-enriched membranous condensates that are often found docked on the cytoplasmic surface of germline nuclei. Germ granules are present in the germline cells of many, if not all, animals, including worms, flies, and mice. Germ granules are thought to act as organization hubs for post-transcriptional gene recognition to promote germ cell function. The most famous germ granule is the P granule in C. elegans, as everyone may have heard the name. However, after nearly 20 years of study, people found that the C. elegans germ granules were found to be commonly compartmentalized into at least 4 subcompartments. In addition to the P granule, 3 additional subcompartments of C. elegans germ granules have been reported in adult germ cells, including the mutated foci, the Z granule, and the smear foci. Z granule, smear foci, and mutated foci. Distinct proteins have been identified that mark the P, C, M, and S granules of the germ granule. For instance, the PGL1 is a germline express protein that is a constitutive protein component of the P granule. MUT16 is a low complexity protein that marks the mutated foci. ZNFX1 is a conserved SF1 helicase domain containing zinc finger protein that marks the Z granule. And tutor domain protein, smear1, marks the smear foci. Many proteins that are required for small RNA-based gene regulatory network are distributed in these different subcompartments. We first asked whether EGO1 localized to the P granule. The answer is no, if we look at this carefully. GFP EGO1 did not co-localize with an PGL1 tag RFP, which is a commonly used marker for the P compartment. So we can say they didn't co-localize, perfectly co-localize. WIGO1 is a worm-specific alkaline protein, which is used as another marker of the P granule. We generated GFP WIGO1, mCherry-EGO1. Okay, show here, GFP WIGO1 and mCherry-EGO1. And visualize the co-localization of WIGO1 and with EGO1. Similarly, EGO1 did not co-localize with WIGO1. Therefore, EGO1 does not localize to the P granule. We then further generated fluorescent target proteins for Z granule, mutated foci, and smear foci, and crossed them onto GLP EGO1, and found that EGO1 does not accumulate into the Z granule, not mutated foci, and not smear foci. Therefore, we speculated that EGO1 may form a new perinuclear granule in the germ cells. To further confirm that EGO1 may form a new perinuclear granule in the germ cells, we tested the genetic requirements of EGO1 granule formation. MIP1 and MIP2 are required for the perinuclear granule formation of the P granule. When EGO1 was disrupted, the P granule was depleted. However, the mutation of these two genes, MIP1 and MIP2, did not affect the distribution of the perinuclear GFP EGO1 foci. Together, this data further suggested that the C. elegans RDRP EGO1 accumulated to an unknown germ cell focus. We collaborated with Dr. Mengqiu Dongslab and used immunoprecipitation followed by mass spectrometry to identify EGO1 interactors. Shown here are the top 10 IP mass hits. Among them, EKL1 and DH3 have been reported previously to interact physically with EGO1 and are both required for RNA, indicating that our EGO1 IP mass assay was successful. The complex of EGO1, DH3, and EK1 will be collectively named as the EGO1 module in this talk. To confirm the protein-protein interaction result from the IP mass, we generated the deletion alleles of the top 10 hits. Then we fed each mutant with double-strand RNA and tested whether these genes are required for feeding RNA. MEX3 encodes a KH domain protein that regulates plasmid identity in early C-Alex embryos. Deletion of MEX3 caused unhatched embryos. POS1 is another essential gene required for embryogenesis. Lockdown POS1 also leads to unhatched embryos. Among the top 10 IP mass hits, we found 2 new genes named EGO1 and ERNI1 are required for feeding RNA. The mutation of these 2 genes leads to RNA defective phenotype. To further confirm the requirements of EGC1 and ERNI1 in the Germ9 RNA response, we tested the silencing efficiency of a Germ9 expressed H2B transgene upon GLP RNA. The loss of EGC1 or ERNI1 significantly prohibited the silencing effect of H2B GLP upon feeding RNA. However, a SOMA expressed SUR5 GLP transgene was still effectively being silenced in EGC1 and ERNI1 mutants, suggesting that EGC1 and ERNI1 are not needed for somatic RNA, but specifically required for Germ9 RNA. We then generated GLP and mCRNA targeted EGO1 inductors, including EGC1, ERNI1, EKL1, and DH3. As expected, all of these proteins accumulated to perinuclear foci in germ cells. And all these interactors do not colocalize with the p-granule marker PGL1. So EK1, DH3, EGC1, and ERNI1, they don't colocalize with the p-granule marker. To test if EGO1 and its 4 interacting proteins colocalized to the same germ cell foci, we used genetic crosses to generate the animals expressing combinations of the GLP or mCRNA or target proteins described above. We found that EGC1 colocalized with the EGO1 and EGO1 colocalized with ERNI1 and so on. So we also did image quantification of the spatial overlap in fluorescent signals and confirmed the colocalization of the 5 proteins, that is EGC1, ERNI1, EKL1, DH3, and EGO1. And therefore, we also named them as the EGC complex. We also confirmed that the 2 new proteins, EGC1 and ERNI1, EGC1 and ERNI1, they don't colocalize with the Z granule marker or mutated foci or smear foci. So therefore, we conclude that EGC1 and ERNI1 localized to a new subcompartment of the germ granule, which we named as the E compartment. And the EGO module mainly accumulated in the germ granule subcompartments. We wondered how this E compartment might be positioned relative to the other subcompartments of the germ granule. To address this question, we generated animals express BFP, PGL1 BFP, mCRNA JNFX1, and ERNI1 GLP to simultaneously visualize the P, Z, and E compartments by 3 different colors. We imaged 200 per nuclear foci and quantified the number of times that the P, Z, or E subcompartments was in contact with the other subcompartments. We found that E compartment is nearly always found adjacent to the P. E is also close to P and is sometimes in contact with both the P and Z compartment. The assembly of many molecular condensates is driven by intrinsically disordered or called low complexity proteins. The intrinsically disordered regions are speculated to mediate the process called phase separation or liquid-liquid phase separation. Among the E compartment proteins, EGC1 and NE1 possess contain low complexity region domains, suggesting that the two proteins might help to mediate the assembly of the E compartment. Then we investigated the mutual dependency of the E granule assembly with other polynuclear granules. We first examined the subcellular localization of the E compartment upon the depletion of EGC1 or NE1. In EGC1 mutant, the number of NE1 GLP labeled foci was strongly reduced. Similarly, in NE1 mutant, EGC1 GLP diffused throughout the cytosol and in the nucleus, suggesting that the localization of EGC1 and NE1 to the E compartment was interdependent. And the localization of EGO1, DH3, and EK1 to the germ cell foci also decreased in both EGC1 or NE1 mutants. However, EGC1 and NE1 remained localized to polynuclear foci in DH3. In DH3 mutant, the EGC1 and NE1 still remained to the polynuclear foci, in which the function of the EGO module was severely blocked. So this data suggests that actually EGC1 and NE1 can be a core factor to organize the E compartment, but DH3 can and EGO1 and EK1 may go to the E compartment downstream. In EGC1 and NE1 mutants, the assembly of P granule, C granule, mutated foci, and smear foci are not obviously changed. So that the other 4 granules don't depend on the E granule. On the other hand, the mutation that disrupt the assembly of P granule, C granule, mutated foci, and smear foci don't disrupt the assembly of E granule either. So in these mutants, the EGC1 and NE1 still form the polynuclear granule. Therefore, these granules may have distinct, different genetic requirements for assembly. So we have a short summary here. We think we found a new polynuclear subcompartment and named it as an E compartment. With some data not shown here, we thought that there are at least 7 subcompartments in the polynuclear region in germ cells. To test whether EGC1 and NE1 are required for 22G RNA biogenesis, we sequenced the total small RNAs from wild type, EGC1 and NE1 animals in a 5' end phosphate independent method. 22G RNAs were mapped to the same elegans genome, and the number of sRNAs complementary to each C elegans gene was quantified. Germline expressed genes targeted by 2-fold fewer sRNA in EGC1 or NE1 animals than wild type animals were identified. The analysis identified 1,500 or 1,300 genes whose sRNA pools were depleted 2-fold or more in EGC1 and NE1 animals respectively. sRNA pools altered in EGC1 or NE1 overlapped extensively. Thus, we refer to the sRNA pools depleted in both EGC1 or NE1 animals as the E-class sRNA, and the genes targeted by these siRNAs as the E-class genes. The E-class siRNAs were also depleted in EGO mutant animals. Therefore, and we conclude that EGC1 and anyone regulate the biogenesis and on the stability of a similar comfort of EGO1 dependent 22G RNA. We also compiled the siRNAs from mutated foci. So we sequenced the siRNAs from the mutated animals. In mutated animals, the mutated foci was disappeared. We identified nearly 1,700 siRNA pools depleted twofold in mutated animals. We also called it M-class siRNA. We compared the E-class siRNA with M-class siRNA, that is, and the siRNA generated in E granule and the siRNA generated in mutated foci, and found that the E-class and M-class siRNAs generally target distinct gene cells. They don't overlap. Metagene analysis confirmed that E-class siRNA were generally unaffected in mutated animals that lack the mutated foci, and the M-class siRNAs were unaffected in animals that lack the E compartment. So they are independent with each other. The elegans 22G RNAs can be grouped into two major classes, called CEDA1 class and the WIGO class, based on the underlying proteins to which they bind. Current models suggest that the CEDA1 class siRNAs are synthesized by EGO1, and the WIGO class siRNAs are likely synthesized by the mutated foci localized RDRP, that is RF1. A comparison of our list of E-class siRNA with the published list of CEDA1 bound siRNA showed that the E-class siRNA represent a subset of CEDA1 siRNA. So which is consistent with previous reports. The M-class siRNA are largely WIGO class siRNA, so that binds to agar proteins. Interestingly, the most interesting striking thing actually is here. When an analysis showing that the E-class siRNA mapped to E-class genes revealed that the E compartment promotes specialized synthesis of 5' siRNA, which is predominantly mapped to the 5' portion of the E-class genes. For example, siRNA targeted CS2 or targeted KF7 were evenly distributed in wild-type animals. So siRNA target evenly distributed or for the whole gene. However, in EGC1 and NE1 mutants, the 5' and siRNAs were disappeared. But the small RNA targeted to the 3' end are still there. In EGO1 mutant, all of the small RNAs are disappeared. So this results that the siRNA targeting the 3' most of the E-class genes rely on EGO1, but not EGC1 and NE1. And typically, and actually this part typically include the last exon of the E-class genes. Metagene analysis showed that the loss of siRNA mapping to the 5' portion, but not the 3' portion of siRNA of E-class genes, was a general consequence of EGC1 mutant or NE1 mutant. Together, this data suggested that the E-class compartment of the Germ granule promotes a specialized synthesis of E-class 5' and siRNA. Finally, we proposed a model outlining a possible architecture of the elegans Germ granule subcompartments and their potential functions for these subcompartments in siRNA synthesis and germ cell functions. The E and M subcompartments are two interdependent subcompartments of the perinuclear germ granules for 22G RNA generation. This is E here, this is M mutated foci here. E compartment derived 22G RNAs are bound to CEDA1 and WIGO4, while 22G RNAs derived from the mutated foci are bound to WIGO1 and HERD1. 22G RNAs mapping to the 3' regions of E-class mRNAs are likely produced by the EG1 module in the cytosol. The function of the gene small RNAs and biogenesis of small RNAs in these 7 subcompartments in the perinuclear granules actually is very interesting as they are studied. So finally, I want to thank everyone in my lab. Especially, I want to thank 2 amazing graduate students in my lab, Dr. Xiaolin Wang and Dr. Ke Wang, who have done this amazing work, very interesting and amazing work. So I want to also thank everyone in my lab for their suggestions and help. And I want to thank my CGC and the National Biological NBRP in Japan for their mutants. And I thank my collaborators, Scott in Harvard, Erica in Cambridge, Meng Qiudong and Shouhong Lives, and Chen Qi and Pan Pan at USTC. Thank you very much. I would like to take any questions.
Okay, thanks, Shouhong. Okay, let's see. Okay, seems there's no question. So okay, I guess we kind of finished today's seminar. So thanks everybody. So I think that's it today.
Okay, thank you.
Thank you.
Thank you, everyone.